Can we regenerate beta cells to treat diabetes? This question has intrigued scientists for decades, and a new study may hold some answers. Today, we're diving into groundbreaking research on a compound called Harmine and its potential to restore beta cells in diabetes. Building off of previous work that showed Harmine is able to increase beta cell proliferation, this new study paints a clearer picture how it's getting that done. Whether you're familiar with diabetes research or just curious about new treatments, I'll break it down in a way that's easy to understand. Let's get started. So let's start with the molecule Harmine itself. It is a beta-carboline and a Harmala alkaloid. Alkaloids are a class of antioxidant, so it has connections to groups of molecules that have antioxidant effects in the body. They are also tied to the brain. Harmine itself is a monoamine oxidase inhibitor, which contributes to its spiritual and social lore in the ayahuasca community. So you'll see these are three of the greatest natural sources of Harmine. Banisteriopsis caapi is also known as the ayahuasca vine. So this is one of the two psychoactive elements in ayahuasca. For the purpose of this video, we're not touching on that. Harmine itself has medicinal properties elsewhere. Syrian rue is a Middle Eastern shrub, I believe, and the seeds are very rich in Harmine. That's where I source any Harmine that I use in any of the experiments that I have been doing. That being said, Passiflora incarnata is a species of passion flower. And so you can do teas, you can do powderized, encapsulated powders of passion flower, and you can use this to touch on Harmine, albeit at a lower concentration than those found in the rue and the Banisteriopsis caapi. But a very interesting molecule, regardless of where it's sourced, because of how powerful it has been shown to be in beta cell regeneration. So you'll see two papers here. There are far more than just these two. It has been a very hot topic in beta cell regeneration research, but it's very clear that when Harmine is in the system, it is exhibiting a very pro-growth effect on insulin-producing beta cells. Okay, this bottom one here is really one of my introductions to Harmine as a potential substance of interest, because they showed that when you combine Harmine, which I mentioned is a beta-carboline and a Harmala alkaloid, but it also functions as a DYRK1A inhibitor or a DYRKA inhibitor. And so DYRKA is basically a cell process that influences cell growth. DYRKA itself stops growth. We'll talk about that in just a second. But either way, one of Harmine's effects on the body is that it blocks DYRKA from stopping cell regeneration. And so they're thinking that because of this, Harmine is basically taking the brakes off of cell growth. And there are a bunch of different other therapies that you can combine and synergize Harmine with in order to really target beta cell growth. This bottom one here just happened to be GLP-1, and I believe they used semaglutide. They may have used exenatide or extendin. I forget the name exactly, but it is well established in the research for its abilities to regenerate beta cells. And like I mentioned before, Harmine blocks the replication blocker. So in a healthy system, DYRKA, that DYRK1A, is something that prevents cell growth. And you may be wondering, well, in people with diabetes, why would DYRKA be activated? And DYRKA, you think about ways in which stopping cell growth is important, you think of things like tumors and cancer cells. It's really important for the body to know when to start and when to stop cells from growing, because if cells just grow without being stopped, it can turn into a bad thing for the body as well. However, in the case of beta cells and other cell types in the pancreas, when we want more cells, we need a way to take our foot off the brake, cell regeneration. And Harmine is a tool that does this very well. So beta cells they have the brake on, and Harmine takes the brakes off so that the cells can divide. And people have shown in the research in these, let's see if I can get those papers back, in these papers specifically, that cell growth is happening in beta cells, and those new beta cells are producing insulin. Like I mentioned with that important add-on, those initial studies in Harmine were like, this is exciting because yes, we're getting beta cells to grow, but it wasn't just beta cells. So when they inhibited the DERCA and took the brakes off of cell division, all pancreatic cell types were growing. So you were getting more alpha cells, more beta cells, more delta, gamma, epsilon. And so when you think about that in the context of diabetes, that isn't really all that helpful, because part of what makes diabetes is the fact that we have too many active alpha and not enough active beta cells. So if you are increasing all of those, you're not addressing the imbalance, which is where that blood sugar dysregulation is coming from. So we needed a way to quiet alpha cells and increase beta cells to help bring us back to a healthy level. And so they did this by adding a GLP-1, or glucagon-like peptide 1, which prevented overexpression of alpha cells. So it kind of put the brakes on the alpha cells while letting Harmine do its thing in promoting beta cell regeneration specifically. And these are just some graphs from this paper in particular. This is from 2020, I believe, but they talk about how, okay, well, when you introduce Harmine, these growth rates, this is like the percent of cells producing insulin, it increases, right, above the level of just the control in the GLP-1. And then when you combine GLP-1 and Harmine, everything goes up quite a lot. I mean, you're talking 10, 20, 30% increase in those insulin producing cells. And when you're talking about someone with type 1 diabetes, this is everything. And I'll leave you with a cliffhanger here, because the paper we're here to review had some, controversial, but not controversial in a bad way. Just not what everyone thought was coming. But why is this important? In diabetes, beta cells, the cells that produce insulin, are either destroyed, as in type 1 diabetes, or become dysfunctional and die off, as in type 2. If we can regenerate beta cells in a natural way, we're not just managing symptoms, we're addressing the root problem. And combining this treatment with adjunct anti-inflammatory therapies in things like nutrition, gut, and liver health, the beta cells stand to not only regrow, but flourish. Now, beta cells don't exist in isolation. They're part of clusters in the pancreas called islets of Langerhans, which also include alpha cells, delta cells, and more. These cells play unique roles in regulating blood sugar. So in these islets of Langerhans, which house all of these different pancreatic cell types, there are these progenitor cells which have the ability to become any of the other cell types depending on the environmental conditions and signaling. Now, this graph, or this graphic right here, which I reference a ton, I have a little breakdown of it in episode 46, The Secret Life of Beta Cells, on the Diabetics for Diabetics podcast. This is from a paper titled How, When, and Where Do Human Beta Cells Regenerate? And this is a deep dive into all of the different mechanisms by which beta cell mass can increase. And I mean, just look at all of these different cell types. So acinar cells and ductal cells are both exocrine cell types within the pancreas. So they are much more tied to digestion and less so with producing hormones. But they both show a potential to become beta cells. There are the alpha cells themselves. There have been a couple different compounds, GABA being one of them, that promotes the conversion of alpha cells to beta cells. And then you also have enterocytes of the digestive tract and hepatocytes from the liver that, given all of these different environmental and signaling stimulants, you'll notice PDX is an influential enzyme, I believe. I'm gonna fact check that right now. But I know it has insulin in its name. So PDX1 is a protein coding gene, also known as the insulin promoter factor 1. When that gene is turned on and when it is expressed, that is a sign for all of these different sources within the body to promote more beta cells, more insulin producing beta cells. So just really fascinating to see how this differentiation process has ways to self-correct. And it is very dependent on the environment within the islets themselves and within the body that promote these different cell outcomes. And what better way to visualize this than a butterfly, right? So you have these precursor cells, which are the caterpillars, right? There's so much potential to become anything. They receive the signals letting them know that they need to turn into something. All of these different genes, all of these different signals from the body itself are telling it what kind of butterfly it's going to be. As it starts to grow and come out of that cocoon, it knows what it needs to be based on those environmental conditions and eventually blooms into those exact cell types. Beta cells, alpha cells, delta, gamma, you name it. But there is a whole journey to it, and it is all dependent on the environment within the cell to dictate what it is that is needed. Here's where Harmine's potential gets even more exciting. This study suggests Harmine doesn't just increase beta cell numbers, it may also improve the process of differentiation, ensuring these new cells are functional and able to produce insulin without disrupting the natural balance of the other pancreatic cell types. So, let's take a closer look at the research. The study aimed to answer two major questions. Given that we know that Harmine can increase beta cell numbers, how exactly is it doing it? And are its effects sustainable in the time after regeneration? So now it is time to actually look at and break down the paper itself. It is titled Cycling Alpha Cells in Regenerative Drug-Treated Human Pancreatic Islets May Serve as Key Beta Cell Progenitors. And holy moly, there was a lot of really cool stuff going on. So let's go over some of the key findings here. So from the left side, all previously annotated cell types are present in Harmine treated human islets. Okay, so alpha cells, beta cells, all of the other endocrine cell types, they're still there. So this isn't a matter of wiping things out for beta cells to take over. Everything still exists after this Harmine treatment. Cycling alpha cells, and we'll talk more about what a cycling alpha cell is versus just a regular ordinary alpha cell, but cycling alpha cells uniquely fulfill characteristics for Harmine responsive cells. So previous research has shown that there are specific traits for cells that make them responsive or reactive to Harmine and what Harmine stimulates it to do. So these cycling alpha cells are unique in that they kind of fit all of the checkpoints for what make it work well with Harmine. And the Harmine combinations increase beta cell phenotypic markers in cycling alpha cells. So Harmine combinations, we talked before about a GLP-1. There is also another type of molecule. I can't remember the name of it. We'll touch on it in a little bit, but these basically synergistic combinations of Harmine with GLP-1 and this other molecule give these cycling alpha cells beta cell traits like producing insulin. And then lastly, conversion of cycling alpha cells to beta cells, right? So they aren't just taking on traits of beta cells, they're actually becoming beta cells themselves. So this conversion of alpha cells to beta cells is what's contributing to beta cell mass expansion. So on the right side of the screen, we have to be wondering to ourselves, is this whole like beta cell regeneration thing an increase in beta cell numbers, or is this just shape-shifting alpha cells, playing beta cell? Really, really interesting. And so this is the graphical abstract from the study. There were three main interventions. Obviously, the control was one of them. You had Harmine, which is labeled throughout the paper. If you go on and read it yourself, I recommend that you do. It's always referenced as a DERCA inhibitor or a regenerative drug. They don't like to say Harmine itself because of its ties to ayahuasca, in my personal opinion. But and then the combination therapy. So you have Harmine and GLP-1, and then you had Harmine and TGF beta, or transforming growth factor inhibitor. So that TGF beta has very similar impacts on cell growth cycles as the DERCA cascade. Okay, so basically two things that accomplish the same thing in terms of influencing cell growth: the DERCA and the TGF beta. But under each of these main interventions, they measured the quantities of the different islet cell types during these treatments: alpha, cycling alpha, these kind of in-between alpha beta cells that exhibited both traits for alpha and beta. And in the process of measuring the quantities, they also measured the impacts of the interventions on these cell types by mapping out, and this is really interesting, mapping out cellular transitions through different RNA expression. So over time, they measured the different stimulants and growth factors within each of these cells to try and map out what the cells were turning into over time with these treatments, right? So it's just fascinating. We'll touch on it in a little bit. But you can see they took beta cells from donors, they submitted them to Harmine or the combination therapies. They saw more insulin, they saw more beta cells, or what they thought was beta cells based on the production of insulin. But then they realized that it wasn't a true through and through beta cell. It was kind of this hybrid-esque cell type that was able to do things that beta cells were able to do. And so they speculated this may be an alpha to beta cell transdifferentiation or conversion. So I can't get over the fact that this is kind of like flipped everything on its head, because for the longest time we thought, beta cells were the goal. The bad news of this research is that alpha cells were the most responsive to the interventions. And before reading this research through, you would probably think like, oh, damn it, like we wanted this to work on beta cells, not alpha cells. Why are alpha cells the only ones responding to this treatment? But the alpha cells that were reacting with Harmine were becoming beta cells and making insulin and showing signs of transdifferentiation. So we may not be focused on beta cells at all. This may be the alpha cells serving as a foundation to become and exhibit beta-like symptoms. So Just really, really fascinating stuff. And this is some of the figures that they reference in the paper. Up here is interesting. 3A, you have all of the different cell types kind of mapped out in a almost like a conjoined bar graph in terms of the quantities. And so one of the things that they focused on was this cycling alpha. So this purple here on this end. Obviously, you'll notice differences in alpha abundance. So there is a little bit more alpha here. Beta, obviously, we're really focused on what's happening in that beta area. And then there was also the alpha beta 2, this kind of in between. But for real, the only ones we're interested in here are the cycling alpha, alpha 1 and alpha 2, to be honest, because they all show the ability to turn into cycling alpha, to turn into beta. But just really interesting to see the impacts that these drug therapies have on sheer cell numbers. And then this is one of those, the maps mapping out through our different RNA expressions, what cell types, what direction these cell types are moving in terms of what they are transdifferentiating into. And so cycling alpha seems to be the greatest source of beta-like activity after the introduction of Harmine. So you'll see all these little arrows here kind of pointed in the direction of beta. They're thinking that, based on the paper, that alpha, whether alpha 1 or alpha 2, is kind of starting to become this alpha beta 1, this alpha beta 2, depending on, how long the treatment goes on, and they're still not sure of the exact transition, the sequence of how the conversion occurs, but cycling alpha is the one that is of the most interest because it checks, like I mentioned before, it checks all of those boxes for having the traits that make it reactive to Harmine, and seem to be the source of the alpha cells that become beta cell-like. And so I think it's worth looking at more, like what is going on in a cycling alpha cell versus an ordinary alpha cell. So I mentioned those specific traits of cycling alpha cells that make it susceptible to the effects of Harmine. This is a quote from the paper we're talking about here, and it says the most significantly upregulated genes in the cycling alpha cells were related to E2F targets, MYC targets, G2M checkpoint, and mitotic spindle pathways, all of which are consistent with cell cycle initiation and Harmine-mediated inhibition of the DREAM complex. So, first part, E2F, MYC, G2M, and the mitotic spindle pathways. These are all things that are tied to an increase in cell growth, right? This is showing a cell that is in the process of regenerating. So that's good news. Secondly, the Harmine-mediated inhibition of the DREAM complex. You may be asking yourself, well, what the hell is the DREAM complex? So this graphic is from a previous paper in 2022. Peng Wang is a researcher who is included in the credits of the paper we're talking about, but he's one of the leading researchers in this Harmine beta cell topic. But there is a DREAM complex that basically keeps beta cells from proliferating. So there's something that kind of turns beta cells' active form into something quiescent or hibernating, essentially. It's not active, it's not producing insulin, it's just kind of neutral, dormant, what have you. And this DREAM complex needs to be silenced or inhibited in order for a beta cell to start to regenerate. So, in this way, these cycling alpha cells have all of the markers to say that it is in the phases of regeneration, and that it has all of the things that a beta cell coming out of hibernation and into its active form have. So it basically has all of the good switches for a beta cell that is ready to start making insulin again. And again, I can't reiterate this point more than anything. It is just so fascinating to me. We might be searching for alpha cell transdifferentiation more than beta cell regeneration. We have been giving so much of our attention to trying to replicate beta cells that make insulin, when in fact the body's natural mechanisms of restoring beta cell mass may have to do, it looks like it has to do with converting our existing alpha cells into beta cells, which is exciting because it gives the opportunity to not need to have a lot of beta cells to experience a lot of growth. Before, for people who have had type 1 diabetes for a long time and have very small beta cell masses, they thought, oh, the regenerative effort is going to take a long time because we're having to rebuild all of these new cells. When in reality, if it's alpha cell to beta cell, we've all got plenty of alpha cells that are making glucagon all the time. That's why our blood sugars are always on their way up. So if it's a matter of conversion and not starting anew, this can significantly decrease the timeline it takes to actually restore insulin production. So very, very interesting. And just that first point, the Harmine and GLP-1 combination is a kind of a match made in heaven, because it's silencing the alpha cell qualities of alpha cells, while the Harmine is promoting beta cell regeneration in those alpha cells. So you're kind of attacking it from two fronts. You're quieting alpha cell characteristics and boosting that alpha to beta cell conversion at the same time, which is why they saw such incredible results in terms of insulin production after Harmine use. And so another point off of this is that the researchers were a little flabbergasted because this emphasis on alpha cells converting to beta cells was in conflict with some of the previous research they had showing that they had regenerated these new beta cells. But they opened themselves up to the idea that the only reason they labeled anything a beta cell was because it was producing insulin. But now that we know that these alpha beta hybrids can produce insulin, in previous research they may have incorrectly identified these insulin producing cells as beta cells, and it may be these alpha beta hybrids. So worth rehashing some of those old papers talking about beta cell regeneration, because it might be alpha transdifferentiation. What does this mean for diabetes treatment? If these results can be replicated in humans, Harmine could become a tool for restoring beta cell mass, potentially reversing diabetes or reducing its severity. And they also in the paper talked about some of the limitations, and I want to make sure that we identify those as well. These first one is small sample size. Okay, they used islets from 4 human donors. So some people will take this and be, oh, it's only from 4 people, it's not compelling, we obviously need more, we need more. So be it. Okay, we'll take that. I still think the information is totally relevant and worthy of getting excited about and investing in. Secondly, they were performed in vitro, not in vivo. So this was kind of done in a petri dish as opposed to in a live animal or a live human. it offers a lot more control over the conditions that can improve growth rates. So proliferation rates are higher for alpha and beta cells in vitro than in vivo. This can impact the rate of development of the changes seen. Again, worthy of noting. This one's interesting. So number 3, no exact lineage tracing in alpha cells. The technology just doesn't exist yet. So there's no definitive way to trace the exact path or timetable for how and what steps along the way alpha cells take in this transdifferentiation. So is this cycling alpha cells turn into alpha 1 to alpha 2 to alpha beta 1 to beta? Like, what exactly is the path? And, we don't really know how to trace the lineage of these alpha cells that are becoming beta cells. Number 4, they did not isolate the difference in gene expression between cycling alpha cells and ordinary alpha cells. So we're not sure from a genetic point of view how these cycling alpha cells, which are showing all of these pro-regenerative qualities, what make them different from just regular glucagon producing alpha cells. And they don't know exactly how long this cycling state occurs. Was this a snapshot of just some ordinary alpha cells in the middle of recycling? Is there basically an entirely separate subset of cycling alpha cells that are always kind of in this perpetual cycling, ready to turn into something else? They weren't sure, but there are ways to set up research to find that out. 5th, the RNA mapping can be further validated experimentally using more precise techniques. So the flowchart maps of the RNA expression showing where, the cell types that everything can become, that was done computationally. So there are better ways to illustrate how that's happening than the computer model that they used. You could use that to question the validity of some of the information that they were claiming, but I don't think it invalidates it. 6, alpha cells allegedly have no GLP-1 receptors. So how is the Harmine GLP-1 combination therapy working? Is there another player in this game? So I didn't even know this when I read it. Alpha cells having no GLP-1 receptors. GLP-1 has primary impacts on alpha cells. But because they don't know that alpha cells have receptors for GLP-1, how is it having an effect? So they didn't know if something happened to these specific cycling alpha cells, or if there is another variable at play, something that the GLP-1 acts on that then acts on alpha cells. Like, is there kind of like a mediator for the effects of GLP-1? They weren't sure. And then lastly, they have no idea how fast these alpha cells are cycling. Are they actively going through cycles, or are they suspended in this ready to differentiate stage? I mentioned that just a little bit before. But so a number of limitations. We always appreciate them in scientific research because there's always, some slack to deal with when it comes to finding out how definitive what's being addressed and how it's being talked about. But I don't think any of these negate what was found. And I think it shines a very important light on exactly what's going on in this Harmine beta cell or Harmine alpha cell to beta cell relationship. So what does this mean for you? If you're living with diabetes, this research offers hope. While Harmine isn't available as a treatment just yet, it's part of a growing body of research looking at ways to address diabetes at its root by restoring functional beta cells in a non-intrusive way. In summary, Harmine is an exciting discovery with the potential to revolutionize diabetes care by promoting beta cell regeneration and differentiation. While there's still much to learn, studies like this bring us closer to understanding how to restore beta cell function. If you found this video helpful, don't forget to like, share, and subscribe. We cover the latest diabetes research to help you stay informed and empowered. Thanks for watching, and we'll see you in the next video.